So we're starting chapter 13. If you see these, I recommend you not eat them if you want to live. If you read I like reading the little things they have at the beginning of the chapter. They're kind of interesting. These are death cap mushrooms that have a very powerful toxin called alpha amanitin that I actually knew about before I knew what a death cap mushroom was. Because going to graduate school for molecular biology, one of the tools that's used in a lot of studies is alpha amanitin when you're doing tissue culture cells. Because the way that the death cap mushroom kills you is that alpha amanitin makes it so that transcription can't happen, right? And so people would use purified alpha amanitin, which comes in little vials with a skull and crossbones on them, and dump them on their tissue culture cells to see if certain processes were possible without transcription taking place, right? So there's drugs that keep transcription from happening, there's drugs that keep translation from happening, and when you have tissue culture cells, you can hit it with alpha amanitin and see if it works, hit it with some other drug that keeps translation from happening, see if that works, and then see at what level the regulation is for these. All right, so here's where we're at now. If everybody could put their phones away, please. I have class now. Here's where we're at the beginning of all these chapters. I show the central dogma here, so DNA goes to RNA through transcription, then through protein, through translation. We already talked in the last chapter, the last bit that was on the previous exam was DNA replication, how you get more DNA from existing DNA. And now we're going to talk about the first stage of what's called gene expression. So when somebody says gene expression and they're a biologist, what that means is how the phenotype is determined by the genotype. Now, there are some RNAs that function directly and don't need to be translated into protein, but the classical definition of gene expression is that first you get transcription of DNA into RNA, and then you get translation into proteins. Right? So I've shown a number of different kinds of RNAs here. Unless we talk about them later, again, this is like I'm just showing you what's out there, and this is not a list to go memorize. So the ones circled in red are in both prokaryotes and eukaryotes, and the ones below circled in blue or boxed in blue are eukaryotes only. So in prokaryotes you have messenger RNA, which is the RNA that's going to code for proteins. And then you also have ribosomal RNAs that are structural. Some of them are structural, and at least one of them is a ribozyme. So I talked earlier about RNA being able to fold into a pop-up 3D tertiary structure and functioning as an enzyme. So when we get to, I said earlier, both processing of, in eukaryotes, pre-messenger RNA, removing the introns from it, that's actually, that splicing reaction, the chemistry is catalyzed by an RNA acting as an enzyme. And down here, making the peptide bonds that link amino acids into a polypeptide chain that is a protein is also something that's catalyzed by an RNA. So these are ribosomal RNAs, and we will also talk about transfer RNAs. You guys know sort of what those are, right? So transfer RNAs are the way that you do the translation between the 4-letter alphabet of nucleotides and the 20-letter alphabet of proteins. So we'll get to it in that chapter, but one of Francis Crick's most famous hypotheses, many of which turned out to be true, is the adapter hypothesis. So there has to be an adapter, a molecule that can interact with both RNA, a nucleic acid, and the amino acids of proteins, and that actually turned out to be true. So these guys are going to be required for protein synthesis. In eukaryotes, as we'll see in the next chapter, you get pre-messenger RNA instead of messenger RNA, because in eukaryotes you have to process the pre-messenger RNA in 3 ways. You're going to, and we'll talk about it when we get there, but you're going to add a cap on the 5' end, add a poly A tail on the 3' end that keeps the RNA from being degraded, and you also have to remove the introns, those bits of RNA that do not actually code for protein. Small nuclear RNA is going to be important in processing of messenger RNA. Small nuclear RNA is going to be actually in the spliceosomes, the protein and RNA complexes that do the splicing reaction. And we'll see that just like what I talked about up here, that a ribosomal RNA catalyzes that. That's the same thing I mentioned as well, that you're going to take that. Spliceosome is going to use a small nuclear RNA in order to catalyze that reaction. We will talk about microRNAs as part of, and small interfering RNAs as part of the RNA interference pathway that we touch on just at the end. The rest of these are important, but they're not, we don't have time to talk about them in here. Just to show that there's a whole bunch of different kinds of RNAs, and except for this one right here and this one right here, the rest of these RNAs do a job that is not getting translated into a protein. All right, so remember, we talked about this before, RNA and DNA have the same primary structure in that they're a polymer of nucleotides into a nucleic acid chain. They have a strandedness, a polarity, 5' at one end, 3' at the other end. They both have a pentose sugar that's connected at the 1' carbon to a base. The difference is that, where is it? Up here, instead of thymine, you use uracil for a base in RNA, although U's are just as capable of pairing with A's as T's are. In fact, it's required if you want to get U's put in here, they would have to be able to base pair with T's in the template. And ribose has the 2' hydroxyl that we're getting closer and closer to being able to figure out why that's important for RNA processing. All right. So secondary structure. Primary structure, it's just a chain. We've sort of showed this in the previous chapter. Secondary structure is where you get base pairing just like in DNA, but except for some unusual conditions, generally you don't get like a double-stranded anti-parallel helix like you do with DNA. In fact, you get like these little stems and loops here, and this is just a small excerpt of that. And the stems are formed by base pairing there, and then the loops are not, like CU would not base pair with G, A would not base pair with C, etc. These stem loop structures then will pop up into a tertiary structure. They're shown flattened out like this so that you can see how the which bases interact with which other ones there. So we're going to see these stem loops today, maybe if we get that far, are important for transcription termination. In one way, transcription is terminated in prokaryotes. They're also involved in regulation of transcription and translation that we may or may not have time to get to, and like I keep harping on, the catalytic function, so that RNA, remember, is hypothesized to be the original nucleic acid, and having functioned as DNA, the storehouse of information, RNA, the messenger of information, and protein, the catalyst, RNA used to be able to do all of those, and some of the functions that RNAs have now are vestiges of that earlier time before DNA was actually involved or on the scene. All right, so if you understand how replication works, you kind of understand how transcription works insofar as the chemistry goes. So you will have a template and you have a copy being made. There are some similarities. Polymerization is in the 5' to 3' direction only with respect to the RNA being synthesized here. So the RNA is 5' on the left, 3' on the right. The template here is going 5' to 3' the other way. So you have a template strand and a non-template strand. So the template strand is down here. It's the one that is actually being used to figure out which bases go in next. The RNA polymerase does the same thing that DNA polymerase does. It can catalyze the reaction by removing the alpha and beta, or the two farthest phosphates, away from the 5' carbon, and that releases energy, and then making the phosphodiester bond that adds whatever the next base is to the chain down here. So there's also the non-template strand up here, because unlike DNA replication, only one of the strands is being copied. There's some exceptions that we'll talk about, but for the most part, there's not ever going to be a gene on both strands in the same area. The non-template strand is also known as the coding strand. The reason being, if you read the template strand, or if you read the non-template strand or the coding strand, that's going to have the same base sequence that the RNA will, except that in the DNA, obviously, you get T's, where in RNA you would get U's. So if you want to read through this, if you're trying to figure out what protein that would code for, when we get to the translation chapter, we'll figure out how the DNA gets translated into protein, but you would be reading these bases here going 5' to 3' on double helical DNA, because they would match that copy there. There are some differences between replication and transcription. One I said was generally only one strand is getting copied. The transcription bubble is only transiently open, so as the polymerase moves from left to right on your screen, this DNA in front of it will unwind, the DNA in back of it will rewind, because only a very small amount, I think like 9 to 12 bases of the RNA is ever in contact with the template DNA. The rest is released because you're just making an RNA copy. You're not putting another strand of the DNA on there. And so that bogus homework, if you downloaded it, that had the extra little question on there that said, why are topoisomerases required for replication but not for transcription, was something that I used to ask before I figured out that I was wrong. It occasionally happens. I thought, well, because this opens up over here and closes there, you're not getting much torque there. But what I was failing to take into account is, while transcription is going, sure, there's a transcription bubble here, but there's another one here, another one here, and they follow each other one after the other. So the total amount of DNA that gets opened is enough to cause problems and that supercoiling of DNA. So experiments have been done, and I found a paper after I thought that I was wrong. I actually found a paper where people showed that if you inhibit topoisomerases, it doesn't completely kill the organism, but transcription is seriously inhibited by not having that there, because you can't get as much of the DNA open. All right. So I already said that usually only one strand serves as a template, but it can be either strand. Now this DNA is flattened out. It's not in a helix. It's if you took all the twists out of the helix, so you can look at only one strand here and only one strand there. So with genes A and B, the RNA sequence is going to match what's called the plus strand. What's the difference between the plus strand and the minus strand? The plus strand is the one that's on top when you draw it, and the minus strand is the one that's on the bottom. Simple as that. There's nothing, you know, if you had drawn this, if you take this whole thing and if you had drawn in the opposite orientation, then this would be the plus strand and that would be the minus. So all you have to pay attention to is what strand your DNA is on. So the red gene and the purplish gene over here, gene A and gene C, the coding strand is the top strand. That means the template strand is the bottom strand. So when I draw these, and I've had to fix them on some of the slides, I think this one was correct. When we draw an arrow up and to the right like that, that means transcription starts where this part of the arrow is and transcription goes that way. Even if these DNA strands were not marked, well, for plus and minus, you'd like 5' here. So if transcription goes that way, that means that you're making this RNA that matches this sequence up here using this down here as a template. And this is the same thing for gene C. Transcription starts here and goes to the right. This is what the RNA would look like, except U's instead of T's. This is the template here. Gene B is different because there's the arrow is here and goes to the left, so this is where transcription starts, and it goes to the left. That means here, this bottom strand, 5' over here to 3' over there, is what the RNA would look like, and this up here would be the template. As I said, there are exceptions, and I might have mentioned this before. You generally, in eukaryotes, while there is a limit to cell size and a limit to nuclear size, it's not a hard and fast limit like it is in viruses. And because the viruses, remember I said we were talking about even bacteriophage, which are viruses for prokaryotes, the capsid will auto-assemble. You throw a bunch of proteins and a piece of DNA into a test tube and they will assemble into that virus particle or bacteriophage particle. And so only a very small amount, there's a limit to the upper level of amount of DNA that can actually be in there. And so in some viruses with very small genomes, you will get genes that overlap in bacteriophage as well. So how do you keep the trains from colliding into each other, right? Here, it's not a problem. This gene is on the top strand, gene A, but termination of transcription occurs here, so the transcription that's using the bottom strand as template ends here. The one that's using the top strand as the template ends here, so they will never crash into each other. But if these genes overlapped, you would get, you know, two trains entering the opposite sides of the tunnel and something bad's going to happen. The way that the bacteriophage and viruses get around this is that one of the genes that's coded for by one strand is used early in the infection process and the other ones will be used later. So even though the two genes overlap, they would never both be transcribed at the same time. And it's not that the virus is smart and figured out how to do that. It's that if that was the case, then that would be unviable and the virus would never be able to replicate. All right, so here is a DNA sequence. If the top strand of this DNA is the template, so if this is the template strand, which RNTP, so that's ribonucleotide triphosphate, so which, you can just say the letter of the base, which base would be the last added to the RNA chain that is transcribed from this template. So if this is the template strand up here, what is the RNA strand going to look like? What direction does nucleic acid always have to be synthesized? 5' to 3' with respect to what? the direction that, not the template, the direction that the new nucleic acid is being synthesized. So if this is 5' to 3' up here and it's the template, then you're going to make a copy of it that looks like what? Just the bottom strand, right? So this would open up and you could pretend that this is gone now. The polymerase would be coming through here, and the RNA polymerase would have to be moving from right to left on the screen because you're going 5' to 3' with respect to the nucleic acid being synthesized. That's going to have to be this way. So the RNA sequence would look exactly like the DNA sequence that's shown on the bottom there, except wherever there are T's here, those would all be U's in the RNA. And I do have to take off for that, so keep in mind. If it's DNA, you got to have T's. If it's RNA, you got to have U's. So what will be the last ribonucleotide triphosphate to be added to the chain then? Just A, right? Because it'd be right here. Simple enough. If the bottom strand was the template strand, then it would be G, because if this was the template strand on the bottom, the RNA being made would be made left to right up here, going that way, and G would be the last one. No matter which strand the template's on, I'm always going to ask you when you draw the RNA on your piece of paper to please label the 5' and 3' ends, and please draw the 5' end at the left. Now, yes, this is going to be going that way if the top strand is the template strand, but I would want you to start over here on the left and write 5' dash CAAU GU CG, just take this and write it in that direction. Because we're writing the RNA from left to right, it makes it easier to read and standardized. So, remember before I said about why you actually need topoisomerases? Here's why. These are sequences of DNA that are being actively transcribed. So you can see at the beginning of a gene you have very small pieces coming off the sides, and at the end they're bigger. Like here's another beginning, up here somewhere would be the end. The little line down the middle going through like this way, or this little line going up the middle here, that's the DNA. The little feeler things, the little brushes coming, bottle brush fibers coming off the sides there, is actually the RNA being synthesized. So one thing I'm going to try to talk about over and over when I talk about gene expression is you have to get a rapid amplification. If this was a prokaryote, this gene from here to here, there'd only be one copy of it in a cell, and you want to make buckets and buckets of protein off of that. So the way that you make a lot of protein off of just one gene is first, each gene is used to make a whole bunch of messenger RNAs, because you can copy the gene over and over and over and over again, and even like here, you're copying it 100 times maybe or more all at the same time. And the second, which we'll talk about during translation, is that then each messenger RNA is going to be simultaneously translated by many different ribosomes. So each gene is transcribed into many RNA molecules. Each RNA molecule is translated into many protein molecules, so you get amplification of your expression at both the transcription and the translation level. And that goes for prokaryotes and eukaryotes, although in eukaryotes there's the little problem of you have to get out of the nucleus into the cytoplasm before you can do translation. All right, so here's drawing from the book. What do we think of when we talk about a gene? So when somebody thinks of a gene, generally they probably think of just this purple part. A gene is some DNA that codes for a protein, right? And this is actually the part that's transcribed here and is going to code for the protein, but there's many other parts of the gene that are necessary in order for this to happen. The first is, as we've talked about with way back when we talked about the yellow allele in mice, is the promoter. So a promoter is DNA sequence that is necessary for transcription to take place, but it is not itself transcribed. As we'll see coming up, the promoter is recognized and bound by proteins that are required to get transcription started or initiated. Then down here, there is always part of the transcribed RNA at the 3' end that is past the last part of the RNA that codes for DNA. So after the stop codon, if you remember that from earlier classes, we'll talk about it later. If not, after the last 3 base word that specifies an amino acid, you get a stop codon that says stop translating here. But transcription goes on after that to a point. And that is the 3' untranslated region, or UTR. If you just want to write UTR, I'll take that. Not shown here, but in prokaryotes and eukaryotes, there's also a 5' UTR. So as we'll see in the translation chapter, AUG, which would be ATG in DNA, is the start codon. So methionine is the first amino acid in every protein, at least to start with. Sometimes it gets cut off later on. But you'll get some sequence in here before the first AUG. And that is 5' untranslated region. So this is one gene. That's what I call a gene, or some people call it a transcription unit. If you look here, upstream and downstream are relative terms like proximal and distal, right? And anterior and posterior and dorsal and ventral. Upstream means in the direction of the promoter. Downstream means away from the promoter with respect to the orientation of the RNA. So if this gene was on the bottom strand, then the promoter would be over here. Everything would be flipped, and this would be upstream to the right, and this would be downstream. So upstream is more 5' when you're talking about the non-template or coding strand or the messenger RNA, and downstream is the opposite direction. As we'll see later on, this in gene speak is always labeled as +1. So the first base that is transcribed is labeled as +1. The last base that is before the end of the promoter there is called -1. So +1, 2, 3, 4, 5, 6, 7, 8, 9, 10 going this way, and then -1, 2, 3, 4, 5, 6, 7, 8, 9, -10 up here, right? There is no 0. All right, so here are some differences. We already discussed one of them earlier, and that's this guy right here, between replication and transcription. So first is there's no primer needed. If you think about it, that would make sense. Where have we talked about primers being needed in the past? With replication, who needs a primer in order to work? The DNA polymerase does, because it needs DNA polymerase 3, DNA polymerase 1. Any DNA polymerase needs a free 3' hydroxyl at a primer template junction. Remember, we talked about that. That's where you go from being double-stranded to single-stranded. The single-stranded is the template, and the last base of the double-stranded part is the primer. And what enzyme was used to make primers during replication? Primase. And what were the primers composed of? RNA bases. You got it. So RNA polymerases don't need a primer. Remember, we talked about that in replication. And so this is RNA, and it's an RNA. You're making RNA off of a DNA template, but you're making RNA, and so you don't need a primer to get started here. Again, it's sort of from the RNA world, they call it, before DNA was there. DNA polymerases are unable to synthesize DNA without a primer, but at the time that DNA was coming in, RNA world was still, the way things were, and so you had a bunch of RNA polymerases that could make primers, and so the DNA polymerase never acquired that ability, and the RNA polymerase called primase had to persist, because without it the DNA polymerase wouldn't work. Only one strand is copied there, and I should have put another word here, that is only a little bit of the DNA and RNA are still ever together. So the RNA being synthesized is almost immediately removed from the template DNA and just exists as RNA by itself. All right, so both transcription and translation have three distinct steps in their life cycle, so to speak. Initiation, which is how everything gets set up and gets started. Elongation, which is once it's started, just chugging along the tracks until we're done. And then termination, when it's time, when you reach the end of the part of the gene that's supposed to be transcribed, how does a polymerase know when to stop? Because in prokaryotes and eukaryotes, there's going to be more DNA way past that, so you need to be able to tell it when to stop. Same thing with translation. The ribosome needs to know when to stop translating. All right, so we're going to talk, you don't have to scramble mightily on this. I'm going to talk a little bit about these things now, but this is like the introductory slide, so we'll go over all these. So the first thing that has to happen with initiation is you have to be able to recognize the promoter, where that is. We're going to talk about sigma factor, which is going to hook up with the RNA polymerase and show it where to go. The DNA has to unwind to form the transcription bubble, just like the initiation factors had to unwind DNA to form the replication bubble at the beginning of DNA replication. And then abortive initiation. So the RNA polymerase has to try and try and try and try again to get started, because usually it's several attempts before you can get enough nucleotides together to get transcription going, and once that happens, then away you go. In elongation, like I said, once you escape from the promoter and you're moving down the line, you're just polymerizing until you reach to the area where transcription has to stop, and then the polymerase, the RNA, and the template DNA all have to release from one another, and the transcription bubble is going to collapse. All right, so let's talk about initiation. Okay. Bacterial RNA polymerase holoenzyme consists of a core polymerase and sigma factor. The core polymerase is over here, and it doesn't look like this. It's just, this is a diagram that can show how it goes together. There are two alpha subunits, so these are 5 separate proteins that each have their own N-terminus and C-terminus. They're separate polypeptide chains that are not connected, but they come together to make this polymerase. There are 2 alpha subunits, a beta and a beta prime, and an omega. Why do we have the beta and the beta prime? They have the same relative look to them, but they are 2 different polypeptides with some of the amino acids not being exactly the same. The 2 alpha subunits, there's only 1 alpha, and there's just 2 of them to go together. So 2 alphas, a beta prime, and an omega. And then sigma factor can come in and join them, and then we'll see after escape from the promoter, it gets released. So the core polymerase is what's going to actually do the polymerizing. The sigma factor's only function is to help the core polymerase find the transcription start site. Sigma guides the promoter to where transcription is going to start. The core polymerase has to be able to transcribe RNA from any piece of DNA, so it can't specifically recognize where to go. That's Sigma's job. Right. So let's look at the promoter sequence, and some of this will look very familiar to you if you have taken microbiology already. So here's +1. Here, this is going to be -1 going up this way. As I said, negative numbers are upstream or 5' of the transcription start, positive or downstream. There is no 0 because +1 is the first one transcribed and -1 is on the other side of it. All right, so if you go 5' to the transcription start site, you get what are very handily named the -10 and -35 consensus sequence, and amazingly, they are 10 bases and 35 bases respectively, 5' to where transcription starts. These two sequences are what the sigma subunit recognizes. So the sigma subunit covers the DNA all the way from -50 way out here to +20 if it was to sit down on there, but it's only recognizing and interacting with these bases at the -10 and -35. That's what shows it where to go. And then when sigma is with the core polymerase, that 2 alphas, the beta prime, and the omega, when sigma sits down here, that positions the rest of the polymerase perfectly to get the transcription bubble opened and have transcription starting right there. If you're from bacteria, if you've taken microbiology, do they still, I think they still do, call this a Pribno box maybe? No, they haven't said that? All right, well I can take that out then. I was a microbiology major way back in the old days, and they held to their own ways or whatever when you're in a department that's all microbiologists. So anyway, if you ever see Pribno box, that's the -10 consensus sequence, so named for the person who discovered it. All right, this picture is not from your book. You do not have to at all memorize it. But we're here to talk about a consensus sequence. So what is a consensus? Who can tell me what a consensus is? What? A survey? Yeah? Thank you, Google. I was going to say, as an example, now this is not going to happen. I don't have the funds, but if I was to say I was going to buy everybody lunch and we had to decide where we were going for lunch, what? Just get you pizza. There would be a lot of, I want to go here, I want to go here, I want to go there, right? But we can only go one place, so everybody would have to, what's the consensus, right? What's the general agreement for everybody? So if you look down, here's a bunch of different genes that are in E. coli, and the actual -10 and -35 sequences, if you look down here at the bottom, this is going to be the consensus sequence. So if you take all the promoters, look what they all have at -10, all have at -35. The consensus is what's the most common. The closer the -10 and the -35 sequences are to the consensus, the stronger sigma subunit will bind to it. So there's a difference in a lot of genes in prokaryotes. Some of them are closer to and some of them are farther away from the consensus sequence. The closer you are to the consensus sequence, the more strongly sigma factor or sigma subunit will bind, and the less you need other proteins to help get transcription started. So a lot of promoters for what we refer to as housekeeping genes, genes that are turned on and under all conditions, those are going to be closer to the consensus. The other genes that are farther away from the consensus, it'll be harder for sigma to bind. It's more weakly bound, and so you need help from other proteins binding to other sequences that we'll talk about later during transcriptional regulation, which is chapter 16, I believe. Yes? I would imagine that these are different for each species, like the telomere, but there's probably, what the exact consensus is maybe different, but like the TATAA is a fairly universal thing. I don't know what the situation is for archaea, but for bacteria, yeah, the absolute consensus might be a little bit different, but it's not going to be, I don't think it's going to be that much different for this. And we don't talk about it in this class, it's saved for molecular genetics, but there are also other sigma factors. So sometimes you'll get a different sequence, a slightly different sequence in these, and a different sigma factor will be able to recognize it interacting with the same RNA polymerase. All right, so here's what happens. Sigma factor, along with the core RNA polymerase, the 5 subunits, comes together. Sigma factor will make the core polymerase, or the core polymerase, along with sigmas, the holoenzyme lands on the promoter. That stimulates unwinding of the DNA from double-stranded to single-stranded, and then you get what I talked about earlier, abortive initiation. So the RNA is held on to the DNA by base pairing, and so even though the RNA polymerase can start polymerizing without a primer there, the first base that's added, there's only one base there, so maximum of 3 hydrogen bonds holding everything together. If you get 2, then a maximum of 6. If you get 3, a maximum of 9 hydrogen bonds. So oftentimes the polymerase will try to start and fall off, try to start and fall off, try to start and fall off a bunch of times. That's what's called abortive initiation. These are usually 2 to 6 nucleotides, but once you get from about 9 to 12 base pairs, and I think 12 or so is in the range of what is stuck onto or base paired with the template all the time. Once you get to a certain number of RNA bases paired with the DNA there, then you can get escape from the promoter and you move on down the line. It's at this point where they show it up here, but it's at this point when the polymerase goes to move away from the promoter, it's done with sigma factor. Sigma factor is released from the core polymerase and floats away to find another core polymerase to guide it to the promoter. So sigma is guiding the polymerase to the promoter, putting the polymerase at just the right spot through interaction with the -10 and -35 sequences. That transcription starts at the right spot, and then sigma is released and only the core polymerase goes off on down the line there. All right, so the transcription bubble is only about 18 nucleotides long. I think like 12 or something is the number of bases that are actually hooked on here. And so the transcription bubble moves. Like I said, DNA unwinds as you're moving in the direction of transcription. It rewinds behind. No primer required. I already talked about that. But remember, that's my wake-up alarm. When I have to use my phone for it. It's very soothing. Anyway, but remember, you saw that picture that had all of the transcription happening at the same time, right? There's going to be another transcription bubble coming fairly close behind, and another one, and another one, and another one, so forth down the line. Because you are only transcribing off of one strand, transcription happens in the direction that DNA is unwinding, and so there is no lagging strand. There is no discontinuous RNA synthesis. It's only continuous synthesis. If you were working on this other strand here, you would have to be making little Okazaki fragments, but because you're only transcribing one strand, there is no lagging strand. And so the polymerase keeps on trucking down to the end there, and then you reach the end. So polymerization has to stop, and then this ternary complex, it's called, has to break up. So it's like tertiary, it's a 3 prefix. So what is the ternary complex? That is the RNA polymerase that's not shown here, the RNA molecule itself, and the DNA template. So in prokaryotes, there are 2 different types of transcription termination. In eukaryotes, there's only one, and we talk about it a little bit later. In prokaryotes, there's row-independent termination and row-dependent termination. Can you imagine what the difference between those are? Yes, amazingly, one of them requires row, and it's not this one. So row-independent termination is really cool because it relies on base pairing, that hairpin formation I talked about earlier. So here comes the polymerase. It's coming down through this DNA, and it's already transcribed this sequence on top here, and then it's going to just transcribe this sequence down here. If you look, these 2 are inverted repeats. So if you start with this A here and that T there, and you move in towards each other, you can see that each set of 2 bases is going to base pair together. And that will actually form, you can see here, this hairpin. So here's the stem, here's the loop. This is the rest of the RNA that would be coming way off here. It's just not shown. And right after, so in the template, right after the second part of the inverted repeat, you see there's a bunch of A's, right? So if you have a bunch of A's, then on the RNA you're going to have a bunch of U's. Does everybody remember, or anybody remember, what the difference between a GC base pair and an AT or AU base pair is? The number of hydrogen bonds, and what's the difference? 2 versus 3. Who's got 2? Right. A and T base pairs have 2 hydrogen bonds. GCs have 3, so they're stronger. And if you look, yes, there are a couple of AU base pairs here, but most of the base pairing in the hairpin is the stronger GC kind. And then right afterwards, the RNA is being held in there by very weak bases. And so what happens is the polymerase enzyme is not shown on this picture, but it's going to be right around here. And when these 2 sequences here snap together to make that hairpin, it pulls against the RNA polymerase, and this base pairing in the stem of the hairpin is stronger than the base pairing that's still holding the RNA to the DNA template, and it just rips it loose. So no enzymatic activity or anything else needed. It's just so row-independent termination is sometimes called intrinsic termination because the termination is intrinsic or as part of the sequence itself. And some genes have this. We'll go through it a little bit. Here's the inverted repeats. There's a string of U's, and we'll step through, and then the inverted repeats form, and oops, wrong button, and out it pops. So it completely pulls loose. All right. So if that was row independent or intrinsic termination of RNA polymerization, then the other example is going to be row-dependent polymerization. So on row-dependent polymerization, you're going to require a protein called Rho. Rho has a similar structure and function to DNA helicase. So remember how DNA helicase works? what's the structure of DNA helicase? What did it look like? It had 6 subunits, and what did it look like? What? A donut. It was exactly a donut. And the whole of the donut went around what? The DNA strand. And so it was a 6 subunit donut wrapped around the DNA strand, and it used ATP energy to push against the double-stranded DNA to break the DNA strands apart. Rho binds to a site that it recognizes in the RNA. Now remember, there's a huge amount of RNA coming way over here, right? So this is the 3' end of what's going to actually be transcribed. So Rho will bind there. Rho is I think it's also a hexameric protein, but it's a donut protein, wraps around the RNA, and just like helicase, it's going to race up 5' to 3' until it hits and bumps against the polymerase there. I don't know why this is in the picture. I am almost certain that there's no hairpin there, and this is an artist's mistake, yet it remains in the current edition of the book. So I'll have to go back and reread and try to figure out. I don't think there's a hairpin involved, but I think what happens is Rho moves up there, pushes, pulls on the RNA, which again is sort of tenuously or very loosely base paired to the DNA template there, and it's going to use its helicase activity to pull the RNA free from the DNA template. And once the RNA is out, so if you look back at the Rho independent, imagine that the polymerase is here too. Either using the hairpin to pull it out or using Rho to pull it out, one way or another the RNA is ripped out of away from the DNA and the RNA polymerase, and once that happens, that makes the RNA polymerase fall off, and then the replication bubble collapses. And then the RNA polymerase, remember, is just the core polymerase. 2 alphas, a beta prime, and an omega, and then it will interact with another sigma subunit and go back and start transcription somewhere else. So again, the sigma functions at the beginning, and I guess you could say it functions here again at the end, but it is not really the end because it's going back around to the beginning. It's like a circular cycle. Okay, so we've talked about similarities and differences between DNA replication and transcription. Now we're going to talk about similarities and differences we've also talked about similarities with transcription in prokaryotes and eukaryotes, but here is going to be a difference. So both prokaryotes and eukaryotes are able to transcribe the DNA multiple times. So you will get multiple polymerases transcribing in both prokaryotes and eukaryotes. But because prokaryotes do not have a nucleus, immediately upon exiting the DNA or the RNA polymerase, the RNA can start to be translated. And so here's the gene in red that it's being transcribed from left to right. You can see some smaller, bigger RNA pieces, but you can see these are not histones. This is not the beads on a string because this is not DNA, it's RNA. Those are ribosomes. Ribosomes are so big that even as an organelle you can see them in an electron micrograph picture, and that's exactly what these are. So multiple polymerases move along the DNA in both prokaryotes and eukaryotes. Multiple ribosomes simultaneously that's not right. This should be both as well. So polyribosomes are polysomes, multiple ribosomes simultaneously translating the same messenger RNA. That's prokaryotes and eukaryotes both. What is only in prokaryotes is what is shown here, which is coupled transcription and translation. So in eukaryotes, there are multiple reasons you can't have this. The first is transcription happens in the nucleus, and RNA has to be exported to the cytoplasm in order to be translated, because that's where the ribosomes are. Second is, as I talked about at the beginning of class today, in eukaryotes, you have to process the RNA. You would not want to start translating it right away because you have to put the cap on it, you have to put the poly A tail on it, and importantly, you have to remove all the introns. You don't want to translate the parts of the messenger RNA that do not code for proteins, right? I need to change that slide. All right, so you don't need to, just like when I showed you all the DNA polymerases, you don't need to memorize all these. I just wanted to show you that in eukaryotes, there are multiple different kinds of polymerases, and they do different specialized jobs, but don't worry about that. In eukaryotes, as we'll see, in addition to having multiple different what amounts to core polymerases, you also have a lot more transcription factors. So in prokaryotes, finding the transcription start site is primarily the job of the sigma subunit. But in eukaryotes, we're going to find out that there are a lot more proteins involved. And here's what we're talking about. So in eukaryotic promoters, again, here's the +1. I put my arrow up here. I do not like where they do this on the bottom going that way, because I always want the arrow to be going away from the strand that's the coding strand, or not the template strand. This is confusing. Anyway, there are more interesting sites that are bound by a more complex array of transcription factors. The most important that we're going to be interested in here is the TATA box. So there's a lot of stuff. So TATAAA, TATA box. It's similar to, remember the TATA that was in the -10 sequence? So this is saved here, but it's more complicated. So for eukaryotic polymerization of messenger RNA and some other stuff, we're going to talk about RNA polymerase II. We're not going to talk about any of these other ones. So RNA polymerase II has a subunit structure for the core polymerase that looks fairly close to what we saw in the prokaryotes. However, instead of having one sigma subunit, you have all these proteins coming together in a big complex that are referred to as the general transcription factors. Why are they general? Because they're used for all of the genes, most all of them. They all have TF2 and then a letter coming after them. So TF2 means transcription factor for RNA polymerase II, and then these are complexes. So these are not individual proteins. The one of them I want you to know about is TF2D. So that's this guy here. The A, B, D, E, F, and H excuse me, are the big complexes that if you're very careful, you can isolate them together. Then if you put them under more stringent conditions, you can break each complex together and find out that it's composed of a bunch of smaller components. TF2D has a protein in it. One of the proteins is called TBP, or TATA binding protein. That's going to be what actually recognizes this sequence here. So TF2D has TATA binding protein and a number of other proteins in together with it. TF2D is the closest analog in eukaryotes to sigma. Although, like I said, there's this whole pile of other transcription factors that are required. These are general transcription factors are what sets the polymerase so that transcription will start at the +1 site. So they're good for most all genes, although there can be some changes in different tissues there. So you don't have to memorize any of these, but I'm showing you that here's the core promoter, like, you know, where you get the general transcription factors set up. 5' to that, you get a bunch of other transcription factor binding sites that will, as the name suggests, allow transcription factors to recognize the DNA sequence. And in some cases, you get a bunch of the same elements right in a row. This will cause more transcription factors to bind and stronger transcription. GC box I worked on in graduate school, it's GC, GGC, CGC, like that. And so that's what I'm saying, it's just a specific DNA sequence. Then here's a bunch of other ones. Cat box is CCAAT, and I think something else after that. Octamer is another one. So these are all, and you can see here's GC box, a bunch of them in SV40 is a monkey virus that was used as a model organism. But look, there's different combinations of these transcription factor binding sites for different genes. I'm just trying to give you the idea that it's fairly complicated, and it's binding of all these transcription factors that helps. And here's the distance away from the promoter. So these can be, you know, 100, 150, 200 bases away from the promoter. And also, there are other sequences called enhancer sequences that are much farther away. But let's go up here to the top first. So if we're looking at the core promoter where the transcription is going to start, here's the TATA box at -25. The first factor that comes in is the only one I'm requiring you to know. TF2D contains as one of its subunits TATA binding protein, or TBP. That comes in, recognizes the TATA box, and recruits with it the rest of these guys, the general transcription factors. Once they are there, they will recruit the RNA polymerase core enzyme and also something called mediator. So we're having all kinds of counseling. You know, we talked about a consensus, and now we're going to talk about a mediator. What is a mediator? If you've ever heard the term. What is a mediator? Right, it's something that's going to communicate between two groups. If you are having a dispute with somebody else, sometimes you get a mediator to talk things over, and therefore, like, you know, it's usually try to have it be like a disinterested party that's not going to be, you know, that's going to be neutral between them. Mediator is a complex of about 30 different proteins. So you can see it's not just a blob like this. There's like 30 different proteins that glom together there, and the proteins that compose mediator can be different depending on different tissues, different time points during development, etc. The job of mediator is to be a mediator or talk between the basal transcription apparatus, the core transcription factors here, and other factors at far away places, and to relay the message from here down to there. So we said this is about the general transcription factors. We're talking about like 25, 30, 35 nucleotides away from the start. The regulatory promoter is stuff that are binding sites that are close to the transcription site within a few hundred bases. This is not to scale. These enhancer sequences can be like 100,000 bases away or something, which is pretty amazing. And so then the DNA has to sort of loop for the activator or repressor proteins at the enhancer or silencer sequences to get down there. And there are also, I don't mention in this class, there are special proteins called architectural proteins that bend the DNA so that this sequence at way out here will be close enough to mediator to interact with it. So shown in the picture here, this is an enhancer sequence. So enhancer sequence, as the name suggests, enhances or increases transcription. They bind to transcriptional activator proteins. So the enhancer sequence is the actual DNA sequence itself, and the activator protein is what binds to it. There are also sequences called silencers that will tend to reduce or eliminate transcription, and they will bind to transcriptional repressor proteins there. All right. So we've sort of seen the structure of the DNA polymerase, how that works. You've seen the RNA polymerase just drawn as blobs. Here's what it actually looks like. So I don't know where everything goes in here, but these two, like, it's sort of like a crab claw like this. The pincher, and the pinchers where my thumb and fingers are is this guy here and this part down here. Those are the beta and beta prime subunits. This is a really cool thing in that, remember, you're having the sequence separated for the transcription bubble. The polymerase is moving from left to right here, and what happens is, as it moves from left to right down the track, it is forcing the double-stranded DNA to become single-stranded. And it does not need helicase to do that, so its own force of action pushes that away. And as the DNA is unwinding, it's kind of hard to see from this picture, but imagine if the polymerase is going this way and the DNA is being fed in here. You can see this part that is in red and has dotted lines. That is not going inside. This is the non-template, or the coding strand. Only the coding strand goes inside. Only the non-coding or template strand goes inside, and it is in the catalytic site. So if I'm going this way, right, left to right, the DNA is unwinding, and the non-template strand is going around the outside of the enzyme. The template strand is going inside. Polymerization is happening. The RNA is coming out down here, and then the DNA template is coming out over here, wrapping around and meeting it. So the DNA separates as you're going like this. The non-template strand goes around the outside, the template goes through the middle, and then they meet on the other side, which keeps the non-template strand from ever getting into the catalytic site and having a problem. All right, so only 2 more slides. We'll finish it up next time.